My program is twofold. On one aspect, I work on ALS, which is the most common form of human motor neuron disease. And on the other side, I use also animal model to study Alzheimer's disease. My focus is really try to understand the brain-body interaction in both disease, because basically these diseases, although they are centrally located, meaning that the neurons die in the brain or in the spinal cord, the cause and also the mechanism might lie outside the brain. The population is aging, as you know, and age is the main risk factor for all these neurological diseases such as Alzheimer's and ALS. So there's unmet need in terms of biomarkers, cure, and also just basically the science per se is interesting. So that's why I'm after this, understanding this disease. My main focus now is on the gut microbiome, its role in protecting ALS, which is the most common form of human motor neuron disease. And this is in collaboration with Lallemand, who provide us with a probiotic that is essential for us to understand how change in the diet, in the lifestyle can influence the progression and the manifestation of this disease. We are really at the basic research level now. We have identified target and pathway, and ultimately what we want to do is to translate like in patient. We have a clinical trial for fecal material transplant, and I know that there's another group that is unrelated to us that use a probiotic, and I think our common goal, I think it's really to drive the basic research to translation for potential clinical trial. We use animal models of ALS, mostly mice, but we also combine that with samples from ALS patients. We have collaborators around the world who collect fecal and saliva material for us, so we can sequence those to determine what are the bacterial content, and then that related to our animal work. So we do everything in parallel. I think the most interesting data that we got so far is that once you trigger a motor neuron disease in an animal model of ALS, and we add back the fecal material from a healthy donor, you can extend the lifespan of these mice. So basically that provides us with a unique opportunity to do translational research. And in that sense, we have a clinical trial that is planned for this year at the University of Calgary. Well, I think the collaboration was instrumental for our progress because they provide us with a probiotic that we would not have otherwise, right? And we made really important discoveries in the sense that it only has an effect on female mice and not on male. So that sex-specific protective effect is really interesting. And again, without Lallemand, we won't be able to achieve this. We usually ask the question that nobody asks, like the sexual dimorphism is a really hard to address because you need to consider the biological sex of the donor and the receiver when it comes to FMT. And so basically when you do the experiment, it's not the double, but it's almost a quadruple, because you have to do the sex match and the sex unmatched experiment. So that is quite unique, I think. And also the other aspect is that we have the clinic of ALS that is nearby. So everything we do needs to be translated into the clinic. So we go back and forth in terms of clinical and basic research. And of course, what really I think makes unique is the people in the lab who do the work, right? They are all unique and they all have the way of thinking and the performing the science. And we are lucky to have a good team. Ultimately, we want to promote the thinking that a healthy lifestyle with a good diet, certain supplements, certain probiotic can basically prevent or slow down some type of disease related to the brain. And I think I know it's a big goal, it's more like a dream, but we have to start somewhere.